The next speaker is Mr. Pawan Kumar. Pawan Kumar is a PhD student in IIT Madras. Talk is SprintGapFiller, efficient gap filling algorithm for large-scale metabolic networks.
That's hello and very good morning, one and all. So I'm here to present my work titled SprintGapFiller, an efficient gap filling algorithm for large-scale metabolic networks. So this is going to be a quick overview of the talk. So I'll begin with a quick introduction about a metabolic network and one of the most widely used method called constraint-based model that is used to model these metabolic networks. And second one is about why there are gaps in the metabolic network. And I'll define the problem statement which we are trying to solve, followed by the algorithm which we are proposing, SprintGapFiller, and the results and conclusion regarding that. So introduction to metabolic networks. So metabolic networks which are present in the biological cells are nothing but few bunch of reactions that comprises of enzymes, metabolites, and key regulatory elements. The main important functions of the metabolic network would be to generate energy, to convert the food to building blocks, and to eliminate waste. And metabolic networks to define a phenotype of a particular cell. One of the widely used method to capture a metabolic network would be a constraint-based model of a metabolic network. And it captures a metabolic network using three different variables: GPR's rules or gene-protein reaction rules, stoichiometry matrix, and the constraints. Gene-protein reaction rules capture the information about how genes are related to a particular reaction, and the stoichiometry matrix capture the relation about how a metabolite is related to a particular reaction. And these constraint defines what is the range in which a particular reaction flux should be in. And once we have a metabolic network which captures a metabolic cell, one of the widely used method to get information from these mathematical model is flux balance analysis, where what we obtain finally is a steady state flux distribution or rate of reaction and rate of each and every reaction occurring in a particular cell. So flux balance analysis is a linear programming based method where we try to optimize or maximize a particular biological objective. So like maximizing a biomass, subjected to two constraints. One is stoichiometry constraint, which assumes a pseudo steady state condition, and the second condition is about the flux constraint, which you have defined before. Like this flux has to be in this particular range. So once we did this, what we have is a steady state flux distribution or rate of each and every reaction in the cell. I would any flux vector that satisfies these two conditions, stoichiometry and the flux constraint, is called a feasible flux vector. Following on to why gap in metabolic networks. First of all, why gaps occur in the metabolic networks, what are the reason for it, and how general people have solved it. First, while building a genome scale metabolic model. Genome scale metabolic model, as the name indicates, it's a metabolic model that scales the entire genome. So it will go till the genome, map the particular genome's sequence to a database and retrieve the specific reactions to it. So once it's done, what we have is a draft model. So draft model is incomplete, and the reasons would be some misannotations or because there will be few spontaneous reactions or transport reactions that will be missed off. So for example, where is the gaps in this draft model? We have a reaction A to D, but we don't have a reaction which produces A or which consumes D. So this model is in particular incomplete. So in general, what people used to do is map it to a particular database model, where a database model is a collection of all the reactions that could be possible in a given organism. And in the database model, give this particular reaction and add minimal number of reaction, so that the in general objective would be so that the model would be able to produce the biomass. So add minimal reaction from the database model given a draft model to get the genome scale model out. The second gap that will occur in context-specific model building. So once we have a genome scale model in the hand, it would be fine for unicellular organisms. But in the case of multicellular organisms, where we have to capture a particular context, like context I defined, it could be a particular cell or a disease conditions. Like if you want a model for a liver cell, or if you want to have a model for a brain cell, that genome scale will not be much accurate because it's a superset. It has all the reactions occurring in a multicellular organisms. At that time, what people used to do is integrate transcriptomics. What transcriptomics have is expression evidence for reactions occurring in that particular context. So once we have this transcriptomic evidence, we again have few reactions which would be part of the model, and again the model will be incomplete or there will be gaps in the model. Here I use the same example. Here again we will not have those reactions which don't have any genomic evidence or again spontaneous reactions will be missing. Here the idea is similar here, but instead of database model, people will be use genome scale model or the superset model and add minimal number of reactions so that each and every reactions that has genomic evidence is shown as expressed. Third gap occurs in the microbial community gap filling problem, where it's similar, but each and every compartment defined here as a draft microbes, and here the universal model used would be few bunch of database model together, and these are core reactions or which we have already evidence, and this is the superset model or universal model which again we'll use, and we add minimal number of reactions to this particular model to make the entire metabolic network consistent. So one common relation between these three different approaches which are gaps that arise in the metabolic network is there is always a universal model which has superset of all interactions or metabolic reactions that could happen in a particular cell, and there are core reactions which are defined. For example, the first case, the core reactions evidence comes from the genome, this particular draft model. These two reactions are core reaction. In the second case, the core reaction evidence comes from transcriptomics. So once universal model is defined and the core reaction is defined, how do we get the minimal network out of it? So mathematically, we are defining this problem statement 1, where the input universal model would have the stoichiometry matrix, the reaction set, irreversible reactions of and the lower bounds and upper bounds, the flux bounds for the given bunch of reactions, and the core reactions will be given as input, and the output should be some flux consistent metabolic model N, and the N should have all the core reactions defined in it, and it should be a subset of the entire reaction network. The equivalent problem statement is to identify block reactions in a given metabolic network. What is a block reactions? I'll take this toy network model to explain it. Like similarly, here we have a toy network with the 5 reactions. Here there could be a free flux flow from E to B. But in the case of A to D, it's blocked because D is getting produced, but it's not getting consumed. So the output, if you want to identify a block reaction in this toy network, it would be like A to D reactions. So we are trying to solve this particular 2 problem statements using the SprintGapFiller approach. The SprintGapFiller algorithm, the entire is shown in this particular slide. So apart from the initialization given the input, what major the SprintGapFiller takes helps of 3 different LPs: LP forward, LP reverse, and LP minimal. So this particular algorithm, in case if anyone is interested in this mathematical details, I can explain it later. But what is the aim of these 3 LPs? I will just explain now. For example, in LP forward, the job of LP forward is to get maximum number of reactions that has flux in forward direction. So for example, in the given toy network, I define these reactions which are highlighted in black color as core reactions. If we apply the LP forward, it will try to maximize this flux in forward direction. So we'll have 1 in the particular core reaction. And LP reverse does the same, but in reverse direction. So here we are trying to maximize the number of reactions that have flux in reverse direction. So in this case, for the same given set of 2 core reactions, the flux would be in reverse direction, -1 here in this case. So once we have these 2 flux values, what we have to do in the third step would be to have a convex combination of them. Here the highlighted boxes are just the core reactions, and the convex combination would end up in a flux vector, and it can be shown that the convex combination of 2 feasible flux vector would again end up in a feasible flux vector. So once we have this feasible flux vector, the main aim of getting this feasible flux vector is to have non-zero flux through the core reactions. Once we have that, we can use the LP minimal, which constrains a non-zero flux through the core reactions and minimize the summation of absolute values of the fluxes. Here we have just shown one formulation, but we do have other formulation with respect to mixed integer linear programming or MILP, where it would be minimize the summation of norm of the reactions. SprintCC algorithm does the same, but here we will not have this LP minimal because we are not aiming at finding what is minimal subnetwork here, but we will try to re-implement this LP forward and LP reverse in a repeated manner in such a way that all the reactions which are unblocked could be identified. So these are the first results which we got with respect to getting a minimal consistent model. FastCore and SwiftCore, which are shown here, are existing algorithms which does the same job. And to obtain these results, we have used this Recon 3D, which is a human metabolic network. And for any context-specific model building, given a genome scale model, the input would be some core reactions. So here we derive these core reactions in a random manner. So for a given random set of core reactions or random selection of core reactions, what would be the runtime and what would be the number of solved LPs? So can we improve the speed in than the existing algorithms or what? So using the proposed approach, we can show that the runtime in seconds for SprintGapFiller is faster than FastCore, and it is faster than SwiftCore for if the number of core reactions is less. And there is a disadvantage in SwiftCore saying that it can't handle positive lower bounds. So in that way, SprintGapFiller, which can handle both positive lower bounds and other reversible constraints, is faster than if we take to only FastCore and SwiftCore, which can handle both the positive lower bounds and other bounds. So SprintGapFiller is faster compared to the FastCore. And similarly, in terms of number of LPs used, SprintGapFiller uses lesser number of LPs than the other two approaches. The second results is with respect to find the block reactions in a given metabolic network. So the approach is similar, where we have chosen a random subnetwork of Recon 3D is given and see if which algorithm perform better in identifying the block reactions in a given models. So here again, if you want to show this, SprintCC does the job, which is in the order of from 0 to 3 seconds. But for FastCore, it reaches up to 100 seconds, or even for SwiftCC, it reaches up to 120 seconds. And in terms of number of LPs used, it again uses some 3 LPs to identify all the block reactions. And FastCC uses around 4,000 LPs to identify all the block reactions. Now, next results are with respect to gap filling in Agora models. Assuming that Agora models are some ideal models. Actually, Agora reconstruction is a reconstruction which has around 7,000 metabolic models or microbial models in it, and these are semi-manually curated. So we are assuming that these are ideal models, and we manually remove some 30% of reactions and see if the SprintGapFiller is able to recover how many number of reactions from the database and adds into the final microbial models. So in the x-axis, these are 3 different formulations which I talked about. One is the LP minimal and MILP minimal, and DC is the difference of convex function, which is a newly proposed approach for solving cardinality optimization method. And with respect to true positives and false negatives, all the 3 approaches perform similarly better. But when it comes to false negatives or true negatives, if we use L0 norm minimization or MILP, it has a better performance compared to the other 2 methods. So with respect to conclusion and future work, we have shown that SprintGapFiller is the fastest algorithm to get a minimal subnetwork, and SprintCC is the faster algorithm to find all the block reactions in a given metabolic network. And L0 norm shows better performance than L1 norm minimization. Currently, we are working on SprintGapFiller for single cell model. So since we have shown that the SprintGapFiller has a better time, or it uses less time to get a minimal subnetwork, where it would mostly be useful would be a single cell model, where we have thousands of transcriptomics or omics data which has been sequenced. And once it has been done, why can't we build those 1,000 models in a faster way and analyze it? Second work which we are currently parallelly doing is about microbial community gap filling. In general, genome scale model of microbial building, what automated reconstruction tools does is like assume some minimal media or complete media and build the model. But microbes mostly exist in communities. At the time, the media would in general be a community. In a given media means a particular metabolite that goes in and out of the cell. But here, a metabolite could be from some other neighbor microbes. So that's why we hypothesize that if you gap fill, taking the community into account, the results would be much more better. Future work is about multi-tissue context-specific model building. Till now, people have looked into how to build a context-specific model for a given context. So owing to its computational efficiency, we are trying to use the SprintGapFiller for a multi-tissue context, where in a given time, would we be able to build a multi-tissue model given the Recon 3D or whatever the genome scale model as input. And similar thing would be applied for modeling the host microbe interactions also. So, for example, in the microbial community gap filling, where we define the media to be the neighboring microbes, here the media could be the host for a microbe or could be a microbe for a host. So, if we gap fill with respect in this manner, we hypothesize that the models will be more accurate. Hence, more number of reactions will not be get added. Finally, I would like to acknowledge my supervisor, Dr. Neerav Bhatt from IIT Madras, and other supervisor for this work is Professor Radhakrishna Mahadevan from University of Toronto. And other students who are working parallelly in this project is Subhashri Sridhar, senior PhD scholar from our lab from IIT Madras, and Noor Al-Smidi from University of Toronto. And thanks IBSC and MyTax for funding. And I would like to thank IMSC and the organizers for giving me this opportunity. Thanks.
Thank you. Really nice work. I think you showed how fast the algorithm is. Have you checked the quality of the models, like in predicting fluxes or gene knockout essentiality? Like,
so first thing is, let's say if R-score and Swift code does a better performance. So we have compared with respect to how many reactions they're capturing. So it was around 95% similarity was there with respect to them. And finally, we are looking into this DepMap data where they have transcriptomics for cancer. So currently we have built models for all the transcriptomics, and Subhashri is currently looking into whether the known cancer reactions are recorded in the final model or not.
I think that would be really impactful.
Yeah. Ignorant question. So when you say that SprintCC is doing better than standard method, you mean by flux variability analysis?
Flux variability analysis.
That is, how do you compute otherwise blocked reactions? It is faster.
It is faster. Actually, existing faster algorithms were these two. One is FastCC and SwiftCC.
Okay.
Which, if you want to use flux variability analysis to identify all the block reactions, the number of LPs we would solve this will be around 2n plus 1, where n is the number of reactions.
So, okay, so this is much faster.
This just uses 3 or 4 LPs for entire metabolic network.
So the other question is, all the gap filling tools assume that the blocked reactions has to be in the final model. Are there any existing tools, because I am not following the literature, where you may think that the blocked reaction is actually a misannotation and we should take it out?
In gap filling existing methods, there are both the type of existing methods also. Like we will try to have the universal model a consistent one. So once one person wants to not have any block reactions, he has the liberty to add as much as reactions from the universal model. There are few methods which will not consider them, like where people would mostly focus on just have some flux through biomass reaction alone, that the block reactions would be remain. But in our case, we are giving the freedom of defining the core reactions. So if you want to define the core reaction as just biomass reaction, you will end up having block reactions in your model. If you want to define all the core reactions to be all the existing graph reactions, then we will end up having a consistent model which will not have any block reactions.